everyone, and welcome to Chapter 1 Cell Structure. This is part 1 of this chapter, and just to give you an overview, there are 3 parts. Number 1 is microscopy, which is all about microscopes. Number 2 is more about calculation, specifically magnification, and basically how we are using microscopes to measure cells. And number 3, there is cell structure and function, and this part you should be a little bit familiar with because you have learned some basics in IGCSEs or O levels, but right now you'll be discovering more of these things in much more detail. So let's start with the first part, that is microscopy. Now, in microscopy, there are 3 main microscopes that you are going to be learning. Number 1 is the light microscope. Number 2 is the electron microscope. And there are 2 types of electron microscopes here, transmission and scanning, that I'll be hearing about soon. Now, before we go into the specifics for each microscope, we must first look at some terms. Now, first you must understand that electromagnetic radiation would be involved here. There is electromagnetic spectrum, which means that there is a range of different wavelengths that can be involved in making microscopes. And in the case of the microscopes we'll learn about, that will be light microscope, which uses light, visible light, as a source of electromagnetic radiation, and there's electron microscopes, which uses electrons and how it reflects off different specimens to visualize the image of a particular specimen under the microscope. Now, other than this concept right here, we also need to establish magnification and resolution, because this is a common misconception that a lot of students have, okay? They think that magnification and resolution is the same thing, but it is not. Now, let's look at the definitions. Number 1, magnification is the number of times an image is enlarged compared to the actual size of the object. So if you say that an object is 10 times enlarged, this means that the image is 10 times the size of the actual object. Right, so that's magnification, and usually an X sign or time sign is placed in front of a number to show how many times it is enlarged. So under microscope in the lab, which is the standard one, in Cambridge centers, exam centers, that would be times 10, that would be times 40. That's on the microscope, okay? So that's magnification. How about resolution? Resolution is the ability to distinguish between 2 points clearly and separate. This is quite interesting. What it's saying really is basically that, hey, when there is 2 points, pardon me, when there is 2 points, perhaps like between this and this, right? To distinguish these 2 points would be the resolution. If there is a high resolution, you would be able to see these 2 points as separate from the background and as separate from each other. Now, if there is a lower resolution image, right, maybe there are 2 points, but you cannot distinguish them from one another because they are so close together. It's not clear enough an image to be able to see the difference between 2 points. If there's an even lower resolution, you might not be able to distinguish this point from the background. So that's why it's defined this way: ability to distinguish between 2 points clearly separate. We usually express the units of resolution in nanometers, and this is something we'll look at in a moment. We'll look at it right after we clarify this magnification and resolution thing. Now, an increase in magnification does not equal to an increase in resolution. Let's look at this image, for example, right? This is magnification increasing in each image. At first, it looks quite far away. Once you enlarge your picture, you know, maybe using a camera, it seems nearer, but it's in fact, the image has just been enlarged up to 20 times. Now, if you use a phone camera, you'll be quite familiar with this concept because as you zoom in, maybe the image becomes more blurry. So basically, you have increased the magnification, but the resolution of the image remains the same. right? It did not improve as you magnified the image. The image is not clear, especially if you magnify it a lot, correct? Let's take another picture as an example. This is an image under a microscope of high resolving power. And you can see a lot of detail. You can see there are like little spots here and here. You don't need to know what it is. Just need to know that, hey, I can see more detail. You can even see this vague spot here as separate from the background. However, if you look at this picture, which is from a microscope with a lower resolving power, same specimen, same magnification, but it does seem to blur, and you can barely see this point that's supposed to be here as separate from the background. Therefore, we say that this image right here has a higher higher resolution. Therefore, the ability to distinguish 2 points as separate is much higher, whereas this picture here on the right has a lower resolving power. Alright, it's under a microscope of lower resolving power. It has a lower resolution, and we cannot distinguish this point as separate, as different from the background per se. So yeah, that's magnification and resolution. Now, what determines resolution? And I kind of gave you a spoiler just now. Basically, it's the electromagnetic spectrum. It's what wavelength you choose to use in your microscope. The shorter wavelength used will give you a higher resolving power. Now, wavelength of visible light is longer than of an electron. And because the wavelength is longer, this would be a lower resolving power. Okay, so visible light, longer wavelength, therefore light microscope would have a lower resolving power because the ability to distinguish between 2 points is lower. So even if you magnify it by a lot, the resolution will not improve. The image will look blur when you zoom in a lot. Yeah. Now, how do we describe resolution? As we said just now, the units are in nanometers, right? The max resolution, by the way, is the shortest distance between 2 separate points. And the max resolution is half the wavelength used. Okay, so the longer the wavelength, aka visible light, longer wavelength, means larger minimum distance. 2 points need to be further apart to be viewed as separate. Therefore, this is deemed as a lower resolution overall. Okay, what am I talking about? Let's look at an example in like numbers term, right? So max resolution again is the shortest distance between 2 separate points to be seen as separate. Now, if max resolution equals to half the wavelength used, and the wavelength of visible light is around 400 to 700 nanometers, there's a range there, then the max resolution of a light microscope will be half of that because of this formula here, right? So its max resolution will be 200 nanometers. What does this mean? This means if this point and this point, right, to be seen as separate, it must be at least 200 nanometers and above to be seen as 2 separate points. Actually, in real life, to be seen as separate from the background, the object needs to be kind of larger than 200 nanometers. And if the object is closer or smaller than 200 nanometers, then these points cannot be distinguished and separate. In fact, it will not be visible at all as separate from the background. So for example, a ribosome. Right, ribosome is 25 nanometers. It is the smallest organelle of the cell. You do need to remember that number, by the way. Ribosomes are 25 nanometers. So when you look down a microscope, will you be able to see it? And the answer is a no, because 25 nanometers is way smaller than 200 nanometers, and it cannot be distinguished from the background, and therefore it is not seen. Now, a nucleus, however, is around 1 micrometer. That's 1,000 nanometers, and therefore it can definitely be seen under a light microscope. So yeah, I hope you get the idea of a resolution now. Okay, now that we have established what magnification is and what resolution is, we can talk about the details. What is a light microscope? What does it use? Source of electromagnetic radiation would be visible light. This is how a standard microscope looks like in the lab. I hope you would have a chance to use it if you go to a proper school. Right? If you don't, I think there are facilities out there for you to be able to rent labs. You need to get your hands on one in order to practice. Anyways, the wavelength of electromagnetic radiation here is approximately 400 to 700 meters, and it has much lower energy, okay, much longer wavelengths than the electrons, and the light is mostly focused using mirrors and glass lenses to direct it. In this case, the light is directed upwards and into the lens and into your eyes. Now, this results in a picture that you see through the eyepiece, which is here. Now, the highest magnification it can go is 1,500 times. However, the one that you use in a microscope usually during the exam would have only 10 times and times 40. Now, at such a high magnification, you will be able to see chloroplasts, you will be able to see nuclei and chromosomes when the cells are dividing. Sometimes you'll be able to see a glimpse of mitochondria, but not very often, honestly, mostly chloroplasts. And yeah, that's high enough magnification to do that. The max resolution is 200 nanometers, which is rather low. This is half the wavelength. So minimum is 400, max resolution would be 200 nanometers. You want the max resolution to have as small as a value as possible to be considered high. And that's why we take 200 nanometers and not 350 nanometers. Smaller, the higher resolution. Smaller the value, the higher the resolution. This is considered quite a large value, so it's a low resolution compared to the electron microscope. Now, what is the advantage? Okay, even though it's not very high magnification, high enough but not very high, even though the resolution is not that great, the advantage is live specimens can be viewed. Okay, you can put live cells under a microscope and you'll be able to see them. And if you stain the cells a certain color, the image can be seen as colored. Okay, or if the chloroplasts are green, you are able to visualize green chloroplasts and green cells under the microscope. And if it's live specimen, you can even observe the cell dividing under the microscope. Definitely possible. Yeah, and of course, it's not here. It's not in the mark scheme. But of course, there's also the added advantage of it being cheap, easily accessible. You can buy it on Shopee and other online websites like Amazon. Okay, it's very easily accessible and it can fit on the table. Okay, so that's a huge advantage there. Again, not in mark scheme. They want to hear live specimens, image that can be colored. And by the way, if you're viewing structures on a microscope, you usually would present the actual size of the organisms in micrometer. That makes sense. Microscope, light microscope, micrometer. Now let's look at electron microscope. The source of electromagnetic radiation here is free electrons. The wavelength, contrary to the previous microscope, is approximately 1 nanometer. That is incredibly short, very high energy. However, it's an electron and it's not visible light. So you can't, reflect it using mirrors and stuff. It must be using magnets. Okay, so electrons, they are negatively charged, right? So you can use the magnets positively charged, negative charge to kind of focus it. And also because it has such high energy, you would want it to be in a vacuum environment in order for the electron to travel in straight lines. what happens when it is not a vacuum environment? These electrons are focused and they are the speed is kind of fast. So if there is air, it's going to create a lot of heat, and that will not good for a microscope. Not good. So yeah, that's the source of electromagnetic radiation. Free electrons. The good part about this is that it has a very high magnification. The highest would be 250,000 times the size. Honestly, the ones on the market probably better now, but this is in your textbook, okay? Now, the mixed resolution of an electron microscope is around half the wavelength of the free electrons, which is approximately 1 nanometer. So max resolution would be 0.5 nanometers, which is very high. The smaller the value, the higher the resolution. Because what is this value? Again, this value is the shortest distance, the minimum distance 2 points need to be at. So if an object is like 0.5 nanometer big, right? Anything above 0.5 nanometers, this object will be able to be seen under the electron microscope. The smallest organelle of the cell, again, is ribosomes, and it's 25 nanometers wide, so it would be definitely very visible under the electron microscope. Now, what's the disadvantage though? Even though it has a very high magnification and very high resolution, the disadvantage is because it needs to be in a vacuum environment, only dead material can be examined. And because it's not using visible light, where light can bounce off and there's a spectrum like a rainbow color, right? But because it's using electrons and the reflections of the electrons are read by computers, the computers interpret these images as black and white only. And but we can use software to kind of color it in to view the image better. But when the computer initially processes it, it would be in black and white. Also, there is a big disadvantage that's again not part of the mark scheme very much, but it's a real life thing. This electron microscope usually takes up an entire room or at least an entire bench space. And again, it's a very sensitive piece of equipment because it needs to be in a vacuum environment. Not to mention that it's incredibly expensive to obtain one. You can Google the market rates for an electron microscope yourself. Anyways, because of the high resolution, okay, my Google just activated. Anyways, what I was saying is, since it has such high magnification and high resolution, when we look at electron microscope images, we usually measure them, express their measurements in nanometers. Just FYI. Now, as mentioned just now, there are two types of electron microscopes. There is transmission electron microscopes and there is scanning electron microscopes, right? Now, transmission electron microscopes, you can see here, it's looking at the internal structure of something, right? Usually these specimens have to be laser cut thin in order for the electrons to pass through the specimen in order to be read. Okay, the equipment kind of looks like this. There is a filament that can discharge electrons, and they are focused using these magnetic lenses, and they have to pass through the specimens. And then the imaging plate at the bottom kind of reads, collects the data. I think it kind of measures the speed of the electrons hitting the plate, and therefore the computer is able to process an image. Okay, something like that. But anyways, the point is, you need for a transmission electron microscope, you need to have the specimens to be cut laser thin in order for the electrons to pass through. Now, scanning electron microscopes, by the way, are built differently. Okay, they are built in a way that has a slightly lower magnification resolution, but to visualize the outside surface of objects. So like this is blood cells, this is Drosophila. Drosophila are fruit flies. Eye is a fly's eye. And these are pollen grains, not like powdery as you thought. It's kind of spiky, good for clinging to stuff. Anyways, yeah, and these are different, and this is how you describe them. As I mentioned, it has to pass through the specimen. If it's a transmission electron microscope, it gives a 2D appearance because you cut it laser thin, and you're looking at it from upwards, up, like it's like a from the upside on the surface. You know what I'm saying? And you are able to see details inside the cell. This is a chloroplast. Look how crazy it looks. Just now in the light microscope image, or we call it light micrograph, you could see like little chloroplasts, like kind of like in a circle, kind of floating around, and they look cute and stuff. This is the internal structure, guys. Look at it. It's like stacks of these are called grana. These are thylakoid stacks, and look at how amazing that looks in up close, right? These are details. These are internal structures. uh, Transmission electron microscopes have such high resolution. You can even look at the membranes within those internal structures here. These are tiny thin membranes. That is crazy. Anyways, that's transmission. For scanning electron microscopes, this is a dust mite, I think, or flea. This is definitely lower resolution. However, it's pretty cool tool. It gives you a 3D appearance. It scans the surface of specimens, so you get really cool images like this. But again, it's in black and white, and usually if you see like colors on these images, they are all CGI. They're all computer generated, and they are all like colored in by softwares. But yeah, those are the light transmission electron microscopes and scanning electron microscopes. All right, before we end, just because you're new to this course, this is a common question, by the way. In the exam, especially in paper 3, they often ask you to list the differences between a light microscope and electron microscope. Now, if they do this in paper 3 especially, you should always present your answer as a table. The first row, okay, row, I emphasize not column, right? The first row usually has marks. You have to label features, okay, and then whatever you're comparing and the other one, so A and B. Okay, that first row has 1 mark usually. And then in the table, you have to be specific in your features, use comparative language, and don't tick across, write present, absent, bigger, smaller, right? They want to see words. Now, usually, yeah, it's 1 mark for the first row, but if there are 4 marks, I do encourage students to write 4 points just in case, okay? Just in case the first 3 are wrong, at least there is 1 mark at the end. So yeah, I'll let you fill that in yourself. I think it should be quite obvious. Magnification is 1 point, source of electromagnetic radiation, there's resolution. You can figure this out. I believe in you. Here's other answers if you want to. Yeah, and you can check it once you're done. But yes, this table kind of summarizes our entire lesson today. These are the differences between light electron microscopes. They do sometimes ask you the differences between transmission and scanning as well, so make sure you know that. And of course, make sure you never confuse between magnification and resolution. That's it for now. See you next video. Bye-bye.